All right, welcome back to another episode of Miss Bioscience, and this week we're going to do something a little different. It's also Microscope Monday, and so I thought today we could do some microscopy together, followed by a little bit of DNA extraction that you can do at home with materials that you find in your kitchen. So we're going to go ahead and get started, but first things first is we want to make sure that we've got our safety materials. So I've got my lab coat, I've got my safety goggles, and I've got my gloves. So I am all ready to go. The microscope experiment or the microscope sample that I'm going to show you today does require some materials. So the first thing you're going to need is some sterile swabs. You'll also need some methylene blue. I'll link the methylene blue that I've purchased from Amazon on the comments. And then you will need a microscope, probably the most cost prohibitive part of this lab. I will link this model of microscope as well. Okay, so we are going to be collecting some cheek cells, and the reason why we're collecting cheek cells is because they are a great source of DNA for extraction. So if you've ever watched CSI or those forensic TV shows, they always use a cheek swab. For one, it's very minimal, right? All you need is this to get that sample. It's also non-invasive, so if you're asking somebody to volunteer a sample, it's more likely that this will be successful than a blood draw. And then you also don't have to have a bunch of officers trained up in phlebotomy because that is a lot more involved task than just swabbing somebody's cheek. So if you want to do this lab along with me or use this protocol, first thing we're going to do is swab our cheek for about 30 seconds. All right, so a good rule of thumb is like sing yourself happy birthday, twinkle twinkle little stars, something like that. So now on this swab, I should have a lot of my own cheek cells that are filled with DNA. Next step is to put this on our slide. So here's my glass slide. I'm going to take my cheek cell swab and I am going to draw that sample onto the slide. And I'm going to make sure that I'm rolling my cotton swab. I want to get as many of those cells on this slide as possible. And you want to make sure they're spread out, because if it's all in one spot, it's going to be a lot harder to see. All right, so you can't see them yet, but the cells are on there. The next thing you want to do is take a few drops of methylene blue and add it to your solution. And what methylene blue is going to do is it's going to take those cells and make them visible. Right now we can't see them, we have to stain them. Roll this around and so that dye hits as much of the sample as possible. Last step is we're going to take a piece of lens paper or paper towel or napkin, and we're going to use it to soak up all of that excess stain, because if we look at this right now, it's just going to look dark. We won't see any sample at all. And you want to gently tap the areas where there's excess stain. You don't want to rub because that will move your sample around. Still not going to look like much until we put it under the microscope, but this sample is ready to go. So the next thing I'm going to do is I'm going to turn on my microscope. I'm going to slide this into place. And when we're looking in the microscope, we always want to make sure that we start with, when we're looking in the microscope, we always want to make sure that we're starting with the lowest objective and working our way up as we get it into focus. Okay, and so now I'm going to be able to show you what I'm seeing from my microscope with some of the video recordings that I'm getting from this. So I have now shown you what those cheek cells look like. And I've explained to you why we use cheek cells for DNA extractions, but we have to go through how to do a DNA extraction. And this is one of my favorite labs because first off, it's your DNA, which I think is really cool. It's something that we normally don't get to see. And second, it's stuff that you have in your kitchen probably right now. So what you will need, okay, so you'll need some Dawn dish soap or shampoo. You will need 3 tablespoons of water in 1 cup. And then in another cup, you'll need 1 tablespoon of either salt water or Gatorade. And this is gonna get swished around in your mouth. So Gatorade tastes better, but all I have today is salt water, so that's what I'll be using. So the first step is to swish that mouthwash salt water mixture. And we're going to swish for about 30 seconds. So again, that's about as long as it takes us to sing happy birthday or the alphabet song or something. Cheers. All right. Gatorade tastes a lot better than salt water, just a heads up. Whew. Make sure that your sample goes back into that cup. We don't want to lose it. So while that's sitting, we're going to make up our detergent. So in this sample right now is salt water or Gatorade. There's also a bunch of your cheek cells. There also might be other stuff, right? Like maybe some leftovers from a meal, or maybe you didn't brush your teeth very well this morning. So there's other things in here that we don't want. And so we're going to go through a couple steps to make sure that what we're looking at is just our DNA. So the first step is that we actually need to even open those cells up to begin with. And so we're going to use a detergent. The way that soap works is it will actually break open the membrane of the cells. That's why we use it to clean our dishes and our clothes and our hair and our bodies and our hands. So this is actually used to extract DNA as well. And in the lab, we use maybe a little more sophisticated detergents, but at their core, they're very similar. And so I have my 3 tablespoons of water. I'm now going to add a tablespoon of dish soap. And you're going to want to give that a really good mix, but you don't want it to get bubbly. So be careful while you're mixing that it doesn't like get all foamy like it does when we do the dishes. Okay, so I have 4 tablespoons now of this mixture because I've mixed 3 tablespoons of water with 1 tablespoon of dish soap. I only need 1 tablespoon of this to go into my sample. So you have enough to do other samples, or you can just clean your dishes with it. Okay, so I've added my tablespoon of dish soap detergent to my cheek swab sample, and I'm going to give this a really good mix now. Again, I want to try to do it gently enough that I'm not getting too many of these bubbles, but I want to make sure it gets mixed well enough. So here is my sample. In this cup is still all of the stuff that I don't care about though. Lots of proteins, maybe some leftovers from lunch, etc. So I have one more step before I should be able to see my DNA. And to do that, I need to go grab something out of the freezer really quick. Okay. So this is 70% isopropyl alcohol, otherwise known as rubbing alcohol. You probably have had this in your house before your first aid kit at a doctor's office when you get a vaccination. So it is super accessible. I got 4 of these at the grocery store the other day for this lab. So what I'm going to do is I am going to pour a tablespoon or more down the side of this cup. It doesn't have to be exact, but I do want it to be down the side, because what I'm trying to create are 2 layers of, I want to create 2 layers of solutions. And I'm going down the side with this ice cold isopropyl alcohol. You don't want to do room temperature. The temperature will also help make sure that we get the striation that we're looking for. Okay, so we see there are 2 nicely defined layers there. So that's all it takes. A couple of chemicals from the kitchen, like dish soap and some rubbing alcohol, and voila, you can see what is normally invisible. So this viscous, kind of sticky, snotty looking material is my DNA. I would encourage you to give this a try at home. It's super simple. The protocol is on my website if you forget the measurements. And it's kind of perfect that we're talking about DNA today because I have an upcoming episode about mutations. And in order to understand mutations, you have to understand your DNA first. We'll see you next time. Thanks for joining me. Bye.